So let's start with our second session. Session is titled Biomanufacturing from CO2. And first speaker of the section works in VTT as a senior scientist. His topic focuses on resource-efficient production of food ingredients and chemical feedstocks by biotechnological CO2 valorization. Please welcome Dr. Michael Lienemann.
Thank you, Ilona. Yeah, I'm the second VTTer today speaking here after Pekka. And thanks, first of all, to the hosts for inviting me and showing such great hospitality. And also my thanks go to Peter Jan for introducing already the food topic. So I will talk a bit more about that. Maybe that's good for the last session before lunch. So I'll trigger your appetite a bit. Not only food I want to talk about, it's also chemicals, but just a little bit. So please be patient then. What I want to talk about more in detail today is about how we basically in VTT do gas fermentation. It's quite different from what is done in Tartu University. And then I will talk briefly about how we use this gas fermentation to produce food ingredients. And then finally, I will touch on the chemical side. So you can use also this gas fermentation to produce precursors for polymers, such as polylactic acid. So here you see a prototype of a sort of headset made at VTT from this polylactic acid resource. You heard already before that the climate change is a big concern and that we have to increase our food production. So there are estimations that between 2005 and 2050, we have to double our food production. And this is a huge task. We believe that this is very difficult to achieve with conventional agriculture. There are several problems to recognize, for instance, soil erosion, eutrophication, excessive freshwater use, and emission of greenhouse gases. And we believe that this gas fermentation is a very interesting technology to produce food with a much smaller ecological footprint. As you see here, this is a sort of prototype, part of a prototype that we built in Lappeenranta in eastern Finland. And you can also produce food then when it's freezing temperature outside, which is, of course, not possible on an agricultural field. It's not just the climate, it's also that if you have soil that is otherwise not suitable for farming, then you can also perform this process there. And you can also do this cultivation not only in a two-dimensional format, you can also stack those. So you can basically cultivate in a third dimension, and this increases further the efficiency of gas fermentation. So what are these foods that we actually want to produce? Here's an example of a single-cell protein product that you can buy already in the shops. It's called corn. This is basically produced by this species. It's a fungus called Fusarium veneatum. And when you compare this to other types of microorganisms here, you see that the composition is quite different depending on which microorganism you choose. For instance, when you take the Saccharomyces cerevisiae, it's a complicated name, it's just the baker's yeast. This has a large proportion of carbohydrates here, which is much smaller in the case of this corn product here. Then we take our lab guinea pig, Escherichia coli. This is the bacteria we usually use for doing microbial experiments. And there you have also very small, in a way, content of carbohydrates. And when you look at this green bar, this is the protein. It's actually quite high. It's over around 50% or higher in all of these microbes. And this is the main application, or the main reason why we are now focusing on microbes for protein production. And the product is then called single-cell protein because protein is the sort of dominating nutrient in this type of organisms. Let's talk some more about proteins. So we have already foods in our shelves in the supermarkets which have proteins. So for instance, chicken breast is a really great source of protein. It's almost 90% is the protein content. And then you have other products which have a low protein content, such as cow milk. So this type of new novel foods that we produced based on single-cell protein, they range somewhere in between those. So for instance, the Australians may know this Vegemite or Marmite, so this is basically a yeast product. This is the corn that I mentioned before. You also have this cyanobacterial product here, spirulina. This actually has also a very high protein content, over 60%. And when it comes to this, now we call the name knallgas bacteria, this is basically bacteria that grow by oxidizing hydrogen. There's a quite large range, so it depends on the cultivation conditions, how high the protein content is of your bacteria. But this is what we are studying at VTT. How do you in a way control the biomass composition, and how do you make it appealing? Once more, so what type of microbes can you use for making this single-cell protein food sources? You can, for instance, feed them carbohydrates. These are heterotrophs. So fungi is one example, such as this corn product. You can use light and CO2. So then you're talking about the cyanobacteria, for instance. This is one example. And this is sort of the approach that the people that I'm working with at VTT is pursuing. So here you would not feed them, you would basically feed them also a gas, hydrogen, as an energy source. And the bacteria would use this energy source to assimilate carbon dioxide into biomass. And as a final example, you have here the methanotrophs, which would use methanol or methanol, both as carbon and energy source. Okay, so we have these 4 different classes. And what does that mean for a process or for a product? So here you have an example of this yeast product. The advantage here is that in a way you can run the process much more efficiently. So this very high energy density substrate allows that these cultivations can be done in at higher densities. But there's a disadvantage with this cultivation of fungi that they may contain toxins, and that they are still depending on the production of this high energy density substrates. When you look at this methanotrophic bacteria, then you have a gaseous substrate. But when you have a gaseous substrate with a high energy, there's a risk that this substrate may actually explode. So this is one risk that you have to consider when embarking on the challenge of doing methanotrophic bacteria cultivation. An advantage here is that they are actually growing quite fast. So this is, again, an advantage for the bioprocessing engineers. The knallgas bacteria case is quite similar. Here the gas is also explosive. But here we have the advantage that we don't have to go to have to transport the methane from some, I don't know, remote sources. We can actually produce the gases, this oxygen and hydrogen as the main energy source inside the reactor or very close to it. You can just basically apply electric potential. Then you can produce hydrogen and oxygen from water. And this has certain advantages. I will go through this further. And finally, we have the cyanobacteria and microalgae. Here you see this spirulina product. This is, of course, interesting when you can just use light energy. That comes almost for free. But the problem there is that it basically happens only in the surface. If you have a deeper reactor, then the cells deeper in the reactor won't get so much light. So there you're working with processes which have quite a low, in a way, density of your cells. And what I heard is also that this algae product have a challenge that they have a bit of a fishy smell. Maybe we'll hear something about approaches to cope with that later on. Just to recap, you can basically organize these 4 different types of organisms based on their productivity and their resource frugality. So the yeast and fungi would be ones that would give you a high yield in a project in a short time, and the algae would be more kind of economically or sustainable. And these methanotrophic bacteria, knallgas bacteria, range somewhere in the middle between those two. You can find basically companies that try to commercialize the single-cell protein production with all these bacteria that I've mentioned. So can, for instance, highlight the NFO Bio. This is a VTT startup trying to make feed for salmon. And we have also Solar Foods that are now producing food ingredients. I think they are already selling this in Singapore. Yeah, just to mention a few. So bioeducation, I'm a biologist, so we like to do experiments. Sometimes we don't have mice, but we have heavy metal fans. So here you see one experiment on a heavy metal festival in Helsinki, and you have two options. You have burgers and you have a vegan substrate. And I think the result is quite clear. So there is some kind of a tendency to prefer one over the other. And I think education helps to sort of cope with fears. So we are basically trying to improve the image of novel foods and also trying to increase the understanding. So at VTT, we are mainly looking into the production cost, safety, and nutritional value of this type of product that we work with. There's also very important part environmental impact, but we have very good experts at Helsinki University, for instance, that deal with this. So a few words still about efficiency. Here's one example of a process that has been established in the 1980s. The company is called Empirical Chemical Industries in the UK. And it's a two-stage process. It consists of a chemical step where you have a CO2 and hydrogen reaction to methanol, and that is coupled to a biofermentation fermentation process. So basically, you would feed this methanol then to this bacterium Methylophilus methylotrophus, and this would then basically oxidize the methanol and grow, and then you have biomass. And if you take this process, here you see one of these reactors that were used back then. They are around 80 meters high, 11 meters wide. And you can basically arrange these reactors on an acre and produce 5 million tons of single-cell protein with this sort of setup. To achieve the same amount of food, you would have to multiply this with 1 million to achieve the same amount of food by soy farming. So this is quite an impressive number, showing what the potential is in terms of sort of resource efficiency of this technology. This company doesn't exist anymore, but it's still a nice example. So this is an excellent paper if you want to read more about this. So let's come back to Finland. Now I want to talk a bit what we are trying to understand about this process here in VTT. As I mentioned before, this is quite interesting, this Knagas bacteria, because you can basically produce the hydrogen and oxygen inside the reactor using just an electric potential. These are the two types of reactors that we usually study. So this on the right, this is exactly this electrochemical approach that I talked about. So basically you apply potential to two electrodes, and on their surface, there's hydrogen and oxygen generation inside the reactor. So you basically only have to feed one gas, which is carbon dioxide. That is the carbon source for the biomass production. The alternative would be that you would feed both substrates, this hydrogen and oxygen, through a sort of gas line, and you purge it into liquid. And the advantage here is that this process is much more efficient if you do it outside of the bioreactor, because you can use solvents that are not optimal for biological growth, but very good for efficient water electrolysis. The disadvantage here is also that, in a way, you're not only producing what you want. You're not having just hydrogen and oxygen, but you're also producing very reactive species that may affect microbial growth. And this is sort of the problem that we targeted in our experiment. I think I discussed that already. I will skip that. So here you see, for instance, a setup of how our reactor looks. You have basically two electrodes, one anode and cathode, where this gas production happens. And as a control, we're having shake flask under an atmosphere containing both hydrogen and oxygen. And these gases basically can enter the liquid through the gas-water interface here. So here are two experiments. And we've taken two bacteria. This is the slide that I skipped before. I should mention this now. Here you have a green box and you have a red box. The green box basically marks all bacteria that we currently consume as foods and that have a gram-positive cell wall. And these red ones have a gram-negative cell wall. What this, in a way, what the important information about this difference for me is that the red ones contain a layer called lipopolysaccharides. And these basically can cause some inflammation reaction. So this is a potential health risk if you consume those. And so we thought that, in a way, we are very used to eating gram-positive bacteria, and the current model organism for Knagas bacteria is actually one of this red group. So we thought, why not sort of exploring more this kind of green group for our food applications? And that's what we did. So we picked 2 gram-positive bacteria. One is called Rhodococcus opacus, and the other one is Nocardiolitis nitrophenolicus. You don't remember. But what you could remember is that this one is something that has not been known to be a Knagas bacterium, but we isolated it in Finland in our labs. And this actually showed quite interesting behavior. So if you look at these squares, this is basically our control experiment, a gas fermenter where you feed gases. So this organism, which is quite well studied, this grows much poorer if you supply the gases through electricity. This is this electricity curve. However, our organism was faring much better in the bioelectrochemical setup. So that's why I thought, like, okay, this may be an interesting kind of production system for food if you don't want to handle large volume of flammable gases. That was Nocardiolitis nitrophenolicus. Okay, so if you have produced your biomass, you have to think, okay, what is the value? Why should I eat it? Is it good? Does it make me healthy? How does it compare to current foods? And one aspect to look at is the amino acid composition. There are essential amino acids that you have to take in from food that the body cannot synthesize. And here you see a list of those. Here again, the 2 organisms that I talked about, Rhodococcus opacus and our bacterium. And then actually you see in our control experiment, it was producing a sufficient amount of tryptophan. In the other conditions with the other species, there was slightly less tryptophan. And then you here use cheese protein basically as a comparison for this analysis. What this means is that in a way, we can now produce biomass, but there may be, in a way, reason to still think about what is a good protein to produce to make it more valuable for you as a food source. Which brings me to the second part of my talk. Now let's talk about what happens when you actually eat this kind of single-cell protein. What you see here is a model of the human digestive system. It contains here the stomach, then you have basically your gut part. And what we can do in the lab, we can basically create reactions that resemble the environment in the stomach and the gut, and then see what happens to your food. Here we have analyzed 5 different samples, 5 different types of single-cell protein. You have the bacterial, Knagas bacteria that I talked before. Then you have a yeast, Rhodotorula. We have already heard about it before. From Peter. But it's maybe a different strain. And then we have sort of 3 different microproteins. This is this corn that I mentioned before. And here you again see something about the composition. They have all protein contents ranging between 20 and 40%. But the dietary fiber is quite different. For instance, here in the case of our Knagas bacteria, you have much more soluble dietary fiber than in the case of the other samples. And you also see that the cells look quite different. So on the bottom, you have a panel of microscopy images. And the left side with our bacteria, you already realize that these cells are much, much smaller than the case of this sort of fungi, for instance. So there are clear differences between the types of single-cell proteins that you can feed. And this picture is supposed to show you what happens then, in a way, in the gut. Here, for instance, we looked at how easy it is for the enzymes to, in a way, cut the single-cell protein down into amino acids. And you see here that if you are basically heating your biomass to make it sterile, you don't want to eat living bacteria usually. So that's why we want to sort of study what happens when you autoclave this biomass. And here you clearly see that the digestibility significantly decreases in these 2 studied samples. We also have here still, in a way, analyzed the production of single-chain fatty acids during this biomass digestion. Here you actually see that there's not so much single-cell fatty acids released from our Knallgas bacteria samples, but there's more from these fungal samples. This is what I had to say about sort of the digestion and food applications. Now we go to this chemical part. Okay, just briefly, this represents a cell, and there's a lot of chemical reaction happening. And basically, we feed it oxygen, hydrogen, and CO2, and all this cell basically converts it to other products. Now I want to talk briefly about sort of lactic acid production. So here, 2 enzymes were introduced to produce lactic acid from this pyruvate. And there's another case where we introduced this enzyme to produce beta-alanine. I'll briefly show you what happened. In the case of beta-alanine production, you should look at this green line. There's an increase of this concentration of this product from CO2, which is nice, but then it decreased again. So the cells started eating up our product. It's again another opportunity to do more research, to study in a way the metabolism of this product in our organism. And in the case of lactic acid, it's quite similar. Basically, we tried 2 different enzymes. In the one case, the concentration was higher, but the production occurred later. And here you have quite comparable production, but the titers were a bit lower. But basically, we demonstrated that you can produce this polymer precursors from CO2. Here, example of a couple of recent articles where these results are presented. And this is the important slide, how we can do this and who helps with this. So these are my brilliant, nice colleagues and our generous funders and our great collaborators. And thank you for your attention. And please, questions.
So thank you. And some questions.
Thank you. Just a quick one about the beta-alanine consumption. So is it a digital thing, or is it meaning that does it kick into action after it reaches a certain threshold, or is it just a question of gradual increase of kinetics?
We haven't studied in a way what's happened there on a sort of metabolic level or genetic level, but you saw that in a way it started to grow faster. So in a way, then the cells sort of got into growth mode, and then they started eating up this beta-alanine.
Yeah, but it kind of like has a bearing how you might actually be able to navigate around it. But that's I guess you are thinking about the same things as I am. So
we can discuss more later. Okay.
So more question?
Yeah. What about this artificial photosynthetic cells, which could be used instead of this photosynthetic bacteria? There are publications where they are claiming that they can get 2 times or even higher productivities in terms of CO2 fixation with these artificial cells.
I'm familiar with the artificial leaf, but I don't know if this relates to the cells.
Yeah, artificial leaves.
Yeah,
but,
yeah.
Okay.
Yeah.
That's exactly the same.
I think artificial leaves are kind of catalysts that use light in a way to, but to be honest, I don't know what the efficiency are they compared to this algae. But we have an algae expert later on. Maybe he can comment more on that. Yeah, thanks.
I have read the papers, so I'm having no personal experience.
Yeah, okay, thanks.
Yeah, thank you. So if no more questions, thank you for a nice presentation.
Thank you.